Okay. So in case you missed the memo, I had at least 2 people email me this morning and begging me to let them take their test on Friday. So if you did not get the memo, the test has been pushed to Friday because of the ice storm. So today will be the last lecture of this section, exam number 3 section, and the exam will be on Friday. Do keep in mind that chapter 15 is solely a recorded lecture. We didn't talk about chapter 15 in class at all. I posted a recorded lecture for that. It has been posted since last Friday, but make sure that you watch it before this Friday. And all of your McGraw-Hill assignments are also due on Friday. There are 4 of them for this section because we only covered 4 chapters. Okay? Anybody have any questions about any of that before I get started? Okay, so we are going to pick up where we left off, and we are in the middle of chapter 13. So we are talking about the adaptive immune system. Oops. And apparently the center projector is the one that's decided to overheat today. So if you all heard it blowing a few minutes ago, we don't have a center projector, I guess. So we left off last time talking about immunologic diversity, and that just means like what we talked about last time, is that each B cell and each T cell is specifically designed to respond to a different antigen. There are 10 trillion, or estimates of 10 trillion different B cells and T cells floating around inside of every single human being. And each and every one is designed to respond to something different. And what happens, and this is just kind of another view of the same thing, are 2 things called clonal deletion and clonal selection. So when these B cell and T cell receptors, so these are this is a B cell receptor, but this also includes T cell receptors, which I don't have a picture of. But when these B cell and T cell receptors are being made, they are proteins, and when they're being made, we get this randomness in the transcription and translation that leads to trillions of different copies that all respond to something different. So there's trillions of different proteins that are made based on the randomness of how they get transcribed and translated during protein synthesis. So what will happen is when these B cells and T cells get created, remember that lymphocytes are both B cells and T cells. B cells and T cells are both lymphocytes, which again are agranulocytic white blood cells. So when these lymphocytes are being created, there will be that randomness in the transcription and translation that we described on the last page. And so each and every one of these B cells and T cells is going to have a different receptor. We've shown them here as having different shapes. They fit, remember that lock and key kind of mechanism that we talked about when we talked about enzymes? There's something very similar here. These proteins are shaped in different ways, and that means that they respond to different shaped antigens. And they make it really obvious here by having tons of different shapes. But each and every one of these cells is going to respond to a different antigen. Now, the first step in this is what is called clonal deletion. And what clonal deletion is it is getting rid of any clones that respond to self. So that's deleting any lymphocytes, so B or T cell, that responds to self. So what that means is if you accidentally created, just in the randomness of transcription and translation, if you accidentally create a white blood cell that attacks your own human tissue, your body's going to get rid of it, or it's supposed to in typically functioning human immune systems. Because we don't want those. We don't want the ones that are going to hurt us. We want the ones that are going to hurt everything else. So that's what clonal deletion is it's the deletion of these self-recognizing lymphocytes. And so what you're left with after clonal deletion is just a repertoire, I guess is the fancy word that they use here. But it's basically just a little army of white blood cells that each and every single one of them is looking out for a different antigen. And that army of white blood cells is going to float around in your bloodstream, and each one is going to be on the lookout for something different. So when things get into your body, these are the things that are patrolling around and looking for those individual antigens. And clonal selection is what happens when one of those white blood cells finds what it's looking for. So in this case, the epitope or the antigen that has entered this human being, right? So this is the little army we've got here of all of the different white blood cells that all respond to something different. But inside this human being, some bacteria or virus or whatever else has entered this human body that has this specific antigen. And this specific antigen binds very nicely with that specific receptor. And so when one of these white blood cells finds the antigen it's looking for, it will differentiate, and they will create basically an army of little clones. Okay, so this is clonal selection. So by having that antigen, it has been, that white blood cell that responds to that antigen has been selected. And so your body will make millions of copies of that exact same white blood cell, okay, that are genetically identical, that are all going to be on the lookout for the exact same thing. Okay? It's like saying, your body has found this thing that does not belong. One of your white blood cells found this one thing that doesn't belong. There is an invader inside of here. We need to get out of it. We need to get rid of it. So we are going to make millions of copies of something that is going to look for that specific thing. Okay. So that is what clonal deletion and selection are. We're going to talk about those. That's kind of a definition of what those are. We're going to talk about them in the context of the immune system in just a couple of slides. Okay. So that was all stage 1. Remember back here, right? I said we were going to break it down into stages. So that's all stage 1. It's how our B cells and our T cells get created, where they mature, how those receptors are made, and how we get these trillions of copies of B and T cells that each respond to something different. Okay, so now we're moving on to stage 2. I just have this up here. We're going to talk about these more in detail in just a minute, but these are the different types of T cells. There are 3 different types, and I have them written down there, but we're going to talk about them as we go through. But I just wanted to have that up as an overview. Okay, so stage 2. Stage 2 of the immune response is the presentation of antigens. And this is how your body is going to respond to things that do not belong. So I think we've talked about it before, but just in case as a refresher, an antigen is just a marker. It's a unique marker specific to any cell. Okay, which is a very generic definition. But that's what an antigen is. An antigen can be anything. It can be a protein, it can be a sugar, it can be a lipid, it can be a glycoprotein, which is a sugar protein, it can be a sugar lipid, it could be literally anything. It's just a marker, a protein or a sugar or a fat or a combination of those things that is unique to a specific cell type. Okay? One type of antigens that you kind of hear a lot about are the ones on your red blood cells that give your blood cells their type. Okay, so I am an A positive, I have an A positive blood type. That means I have A antigens and RH antigens that's the positive. Okay, if you're an AB blood type, it's because you have A cells, or you have A antigens and B antigens. Specific viruses have specific antigens. Bacteria have specific antigens. Okay, it's just any unique marker. But the entire immune system inside of humans relies on our immune system being able to find and recognize those antigens as ones that don't belong. Okay, so the second stage of this process of recognizing something that doesn't belong is something called antigen presentation. Okay, and antigen presentation is what happens when an antigen from a digested microbe is presented on the surface of a cell by an MHC class II molecule. So this is the key here. Antigen presentation only really happens with an MHC class II molecule. Okay. And there were 3 types of cells that have the MHC class II. Remember, everything except red blood cells have MHC class I. It's a couple of slides back. Go back and look if you don't remember. But an MHC class II is only present on 3 types of cells: dendritic cells, macrophages, and B cells. Okay. And so those types of cells that have an MHC class II are known as antigen-presenting cells, also called APCs. So that is going to be the dendritic cells, macrophages, and B cells. So this antigen presentation step is only going to happen when an antigen presenting cell, a dendritic cell, a macrophage, or a B cell, presents an antigen on its surface using that MHC class II protein. Now, the way that this actually works is very poorly understood. And so obviously you don't have to understand it. But what basically happens is phagocytosis happens. We talked about phagocytosis a fair bit. But these cells will phagocytize something that doesn't belong. So this is a microbe, a bacteria in this case. And so this bacteria is getting phagocytized. And then in a process that most scientists don't understand, and you certainly don't have to understand, the cell, the macrophage or the dendritic cell, will take a piece of that microbe, take an antigen, a small piece, one protein or one lipid or one thing from that microbe and put it on its surface as basically a little red flag, I guess. It just takes a little piece of this microbe and it presents it. Okay. It presents a little piece of that destroyed microbe on its surface. And this is your white blood cell's way of saying, hey, this is what I found. I found something that didn't belong. I destroyed it, but this is what it looks like. Okay, it's a little flag to the rest of your immune system to say, this is what is floating around. This is something that I found. And we're going to get to the rest of it in a minute, but what basically happens is that little flag, the little antigen presented on the outside of these cells, works to induce the rest of the immune system. Okay, but this is the little red flag saying, I found this, it doesn't belong, look at it. Okay, and then the rest of the immune system will respond. So that's antigen presentation. And again, remember that antigen presentation only happens with antigen presenting cells, which are the ones that have the MHC class II protein. The only 3, the dendritic cells, the macrophages, and the B cells. So not all white blood cells can do antigen presentation. Only those 3 can. Okay, and then the presented antigens will act to induce other parts of the immune system. All right, so the next kind of part of what's going to happen in this stage 2 is we're going to have the activation of helper T cells. Now, helper T cells, if you go back a couple of slides, helper T cells help to induce the immune system. they help the rest of the immune system do what they need to do. They're the helpers. Okay, so this is how we activate these helper T cells. These helper T cells are floating around in the blood looking for something to fight off, right? They're policemen trolling the streets, looking for things that are not supposed to be there. And the way that they become activated, the way that they become on high alert, basically saying there is danger here, we need to get rid of it, is by becoming activated. And they become activated when they find an MHC class II molecule presenting an antigen. So a helper T cell will bind to the antigen on an MHC class II molecule and become activated. So we have this, and don't get too bogged down in all the details here. I will highlight what you need to know, okay? So this is our antigen presenting cell. Right here, this purple one. This is our antigen presenting cell. It's a phagocyte that chewed up a bad microbe and took a piece of that microbe and shoved it on its surface, presented that antigen with an MHC class II molecule. And once that happens, a helper T cell, specifically a helper T cell, with a T cell receptor that responds to that specific antigen. Okay, remember that each and every T cell has a different T cell receptor, and each and every one will respond to a different antigen being presented. So when the T cell that has the specific T cell receptor that matches the antigen being presented, those two will grab onto each other, right? The receptors grab and bind onto whatever it is they're meant to bind to. So you have the binding happen right here, and then this helper T cell is going to become activated. Okay, that means that it is going to then induce other parts of the immune system, which we're going to get to in just a sec. Another thing that happens in this kind of same stage is clonal proliferation of that specific helper T cell. Okay, because now this specific helper T cell that responds to that one specific antigen has recognized that something bad is happening, that there is something in this body that does not belong here. And so it's going to make tons of copies of itself so that there are more copies to look out for more bad things. It's saying this, it's like putting out like a bee on, I watch too many cop shows. It's like putting out like a bee on the lookout, right? It's like somebody saying over the radio, there's this bad guy floating around. This is what he looks like. This is what they do. Be on the lookout for this specific thing. And that is what those T cells are doing. They're saying, I have found this specific bad thing inside this body. Put everybody on high alert to look for the exact same thing. Okay, so the helper T cells become activated, and they also proliferate. So they make more and more copies of themselves so that they're all activated and they're all looking out for this one bad thing. Okay. And so I just like these little cartoons. This is exactly what I just said. It's just in a little cartoon form to make it cuter, I guess. So, but when a potential threat is detected in the body, dendritic cells or B cells or macrophages, so APCs, eat the cell or debris and carry pieces. So the carry pieces is the antigen presentation. Okay, they carry pieces of it to where the white blood cells are. And they wave it around, and so you can see that this little dendritic cell here has got a piece of this microbe that is pink. And inside the white blood cells, there are all these T cells that have different receptors, right? There's a pink receptor here, an orange one, a yellow one, and a green one. You can kind of see the colors right there. So we have trillions of different T cells that all respond to something different. But the one that responds to this specific antigen is then going to become activated. So the dendritic cell presents the pieces of the antigen to the T cell as evidence of a potential threat. It is saying, I found this floating around in this body. I think it's a problem. Somebody go get rid of it. And so if a T cell has a T cell receptor that matches up with that specific antigen, the T cell will begin to activate. Okay, so this T cell with the pink receptor that matches the pink antigen will become activated. And the activated T cells multiply rapidly, which is that clonal proliferation, forming an immune army that flows out into the body to hunt and attack that specific threat. So now we have a little army of these T cells that all respond to the pink antigen, looking out for that specific threat. So that is antigen presentation. Okay, and then we're going to do stages 3 and 4. Again, this is referring to that original chart on that first page. And now we're going to look at, because this is where things start happening kind of separately. So we're going to look at stages 3 and 4 as it relates to the T cells, and then we're going to look at stages 3 and 4 as it relates to the B cells. Okay, so we're going to look at T cells first and then B cells, but remember that these are happening at the same time. Okay, and so this is where I wanted to talk about those different types of T cells. We have 3 different types of T cells. We already talked about the helper T cells. The helper T cells provide signals that support other immune cells. Okay, so this is the helper guy right here. He became activated, and he is telling the rest of the immune system to do its job. Okay, he's the helper. The cytotoxic T cells encounter antigens that they are programmed to recognize and then release molecules to attack and kill them. So cytotoxic, cyto means cell, toxic means deadly. These actively kill pathogens. But cytotoxic T cells, like you can kind of see in that first picture, cytotoxic T cells won't really respond until a helper T cell tells them to do that. Okay, so they're programmed to recognize. Again, they're programmed to recognize based on this T cell receptor. Because of that proliferation, there are millions of copies of these T cells that are designed to recognize and attack that specific pathogen. Okay, and then the regulatory T cells slow down or suppress the immune response so that the immune system doesn't overreact. Right, when the immune system overreacts, that can actually cause more damage than the pathogen, right? Just like anything else, there has to be a checks and balances system that prevents things from overreacting. So the regulatory ones are the ones that say, hey, are we still doing the right thing? Do we still need to be killing stuff? Is it time to slow down? Did we get rid of everything? And if the regulatory T cells aren't working, that's when you get your immune system attacking your own body. Okay. But that is what happens to the T cells when they are activated. The helper T cells activate other parts of the immune response. Cytotoxic T cells actively kill pathogens, and the regulatory T cells stop or slow down that immune response to prevent overreaction. Now, stages 3 and 4 with the B cells. This is where it gets a little tricky. So what's going to happen with the B cells, and remember this is all happening at the same time in different parts of the body. These are happening simultaneously. So before the B cells can do anything, the B cells have to become activated. Well, how does a B cell become activated? A B cell becomes activated when it interacts with an already activated helper T cell. So just like we said before, the helper T cells help. They induce other parts of the immune response. So in order for this to happen, remember that B cells are antigen-presenting cells. Remember that B cells have an MHC class II molecule. They are also antigen-presenting cells. So when a B cell comes in contact with a bacterium or a pathogen of some kind, it is capable of presenting that antigen just like the other antigen-presenting cells, right? We talked about antigen presentation already. So just like the other antigen presenters, it can see this microbe with an antigen and present it on its MHC class II molecule. And then this helper T cell that was activated in the last stage will come in and recognize that same antigen and then activate the B cell. Now, and when a B cell is activated, it creates two different things. It creates plasma cells and it creates memory B cells. The plasma cells are what we call antibody factories. Now remember that antibodies are just B cell receptors. So plasma cells are cells that are going to produce trillions and trillions and trillions of copies of an antibody that has the same specificity as the antigen being presented here. Okay, so it's going to have the same specificity as the B cell that recognized it. And we're going to talk about what antibodies do in a minute, but antibodies work to kill pathogens. We're going to talk about how they do that in a minute. So a big part of what happens with the B cells is they produce plasma cells, which are antibody factories. They're going to pump out trillions and trillions and trillions of antibodies into your bloodstream. And then those antibodies are going to go out and do what they do to get rid of the pathogens, which we'll talk about in a sec. The second thing that they do is they create memory cells, which is exactly what it sounds like. They are cells that have a memory. It's kind of like putting up, I don't know why I keep trying to relate it to the police department, but it's kind of like putting up like a wanted poster or something like that. The memory cells basically remember pathogens you have come in contact with. Okay. And so what that means, we're going to talk about the immune response in a second, but if you have come in contact with a pathogen before, your B cells are going to remember. And the next time you come in contact with that same pathogen, your body is going to immediately react and get rid of it much, much quicker. Okay, that's how immunological memory works, which we'll talk more about in a minute. Okay, so again, the key here is that you have to, in order for a B cell to become activated, you have to have a B cell that is presenting an antigen and an already activated helper T cell that had to be activated by recognizing an antigen from something else. An already activated helper T cell meet and bind together, and only then do the B cells become activated. Okay, and the reason for that has to do with, it's called immunologic redundancy, which is just a big fancy word, but and the point of immunologic redundancy is that if you're going to waste a lot of resources and you are going to create this huge immune response, like you guys have all been sick before, it does not feel good when your body induces an immune response, right? It creates it uses tons and tons of energy. It makes your body hurt. It uses all of your resources. It makes you extremely tired. It is not fun for your body to do this, to create antibodies and fight off pathogens. It's hard. It uses resources. It consumes energy, right? Again, you've all been sick before. You know how it feels when you have the flu and your body is fighting it off. You want to sleep for 3 weeks and your body hurts and you have no energy, right? It's a hard thing to do. So to prevent your body from doing this all the time, it creates, the only way for B cells to become activated is if there is this redundancy. So a B cell is going to become activated if it finds this antigen, presents it on its surface, and then a helper T cell that has already seen the same antigen somewhere else also comes in contact with that. Is then the immune response truly going to be activated? And this prevents an immune response from happening unless there is truly a threat. Okay, so like if your body comes in contact with a piece of pollen, right, and one B cell finds it and presents that antigen, that's not going to induce an immune response. We don't want that to happen. One piece of pollen, we don't want to induce this huge immune response. And that happens to some people. It's called allergies. That's what that is. But in theory, you don't want your body to over-respond to everything it comes in contact with. If your body created an immune response for every single pathogen and every single antigen that it encountered, you would be sick for your entire life, right? Because you are constantly in being exposed to pathogens, and you are constantly being exposed to allergens and other antigens. And you do not want this to happen every single time you find one. It's kind of like, it's just like a system of checks and balances. It's a way of saying, oh, I saw this one thing over here. Well, I saw the same thing too. Okay, this is really a bad thing. Okay, it's like checking with somebody before making this really big call. Like, I guess the only thing that comes to my mind right now is like with the ice storm, like making the decision to cancel school. That wasn't just one dude that was like, yeah, we'll call school for the whole week, right? No, it was a bunch of people looking at all different kinds of stuff and all the maps and all the weather reports from 16 different states and saying, yes, this is what's gonna happen, we need to cancel school, and coming to that decision together. And that's what your body does here. It's a B cell saying, I think I see a problem, and it's a T cell saying, yes, I think there's a problem too. Let's talk to each other and solve it together. Instead of just one dude freaking out about an ice storm that might not happen, right? Okay, so that is how B cells become activated. And the activation of B cells, again, the ultimate purpose of the humoral response and the B cells is to produce antibodies, which are those B cell receptors that are floating in the blood. They can be released. Right? So now we're going to talk more about antibodies and what it is that they do. So this is what an antibody looks like. An antibody monomer, which is, remember, mono means one. So a single antibody monomer, it's generally in this Y shape. And this Y shape is going to have 2 light chains and 2 heavy chains. So you can see them here. The 2 heavy chains are pink, and they're the ones that are bigger. Okay, so these are the heavy chains. And what I mean by chains is I mean protein chains. Okay, so these are proteins. They have 2 heavy protein chains, so 2 big protein chains, and 2 small ones, known as light chains, which are in green over here. And these heavy chains and light chains are arranged in this Y shape. This is a classic antibody shape. And each individual antibody is going to have these 2 antigen binding sites, which are right here. So every single antibody is going to have 2 places where it recognizes antigens. And that is going to be the variable region. This is the part that's different from every single antibody, right? That's how the antibodies recognize different antigens, because that variable region is variable. It's different. Okay. So the variable region has a unique shape that binds to specific antigens, and every single antibody is going to have a different specificity. It's going to respond to a different antigen. And then the constant region at the bottom is going to have a similar shape in every antibody. It's the part of it that's constant. Okay, but this is considered an antibody monomer. Is this one single Y shape? Now we're going to talk about the different types of antibodies, and there's a lot of information on here. I'm going to highlight which parts of it you have to know and which parts of it you don't. Okay, so there are 5 different types of antibodies, and you do have to know the 5 different types. We have IgGs, IgAs, IgM, IgD, and IgE. There's the 5 classes of antibody. Okay. And you do have to know that IgG, well, we're going to do them one at a time. So IgG is a monomer, which means that IgG exists as just one of those little Y shapes. We're going to look at, IgM over here is a pentamer. It's 5 of them together. But IgG exists as a monomer. It's just one of those little Y shapes is considered a single antibody. Okay. Another part of this that is important is the percentage of the antibody in the serum. Now, you don't have to memorize the exact percentage, but what you do have to know is that IgG is the most in the blood. Okay, so of the antibodies that are floating around in your blood, most of it is IgG. Okay. And another thing you have to know about this one is that IgG is the only one that crosses the placenta. So if a person is pregnant and they have a placenta, IgG is capable of crossing from the gestator into the fetus. Okay? You can cross the placenta into the baby. And so this is part of why they recommend, we'll talk more about this later, but why they recommend that pregnant people get certain vaccines, because the vaccines can make you produce these IgGs, which then cross into the fetus and protect them, specifically for like whooping cough and stuff. Okay, and then IgG also fixes complement. Now, I know you have no idea what that means right now. We're going to talk about it on the next slide. But you do have to know that the only ones capable of fixing complement are IgG and IgM. Okay. And then the other big thing to know about IgG is, in addition to it being the most prevalent in the serum, it is most important during, if you read this bottom part, it is most important during a secondary immune response. Now again, I know you don't have any idea what that really means at this exact moment. We're going to talk about it in a few slides. But just remember that IgG is the most important in the secondary immune response, which is the second time you come in contact with a pathogen. Okay? Next, we have IgA, and IgA exists as a dimer. Okay, so IgA is these 2 little Y's stuck together. So IgA exists as a dimer. And IgA, the only other thing that you really have to know about it, it does not cross the placenta, it does not fix complement. Its biggest thing is that it is found in high concentrations in our secretions. So anything that your body secretes, spit, sweat, vaginal fluids, ejaculate, breast milk, anything that comes out of your body as a fluid is going to have IgA in it. Those are the antibodies. And again, this is how breast milk protects infants from infection, is because IgA is secreted in breast milk. This is also how, this was the very, very first, or some of the first, not the first, but if you're interested in this at all, IgA is how they used to do forensic testing, because IgA is present in ejaculate, in semen. And so they would test sexual assault victims or murder victims and things like that for IgA to see if they could match it to the person it came from. Okay, so that was one of the first ways that they were able to test body forensics before DNA was a thing. I know most of you don't realize that, but DNA testing is actually very recent. Okay. They just started using DNA testing in like the 1990s. Okay, I know that seems like a million years ago to you, but I was born in 1987, so I was born before DNA testing, not that long ago. Okay, I'll try to convince myself. Okay, but IgA is mostly known for secretions. IgM is a very large antibody. It is a pentamer, which means that it has 5 of those individual monomers all hooked together, make one antibody. Okay, and it is produced at first response. So this is most useful during the primary immune response. So IgG is the secondary, IgM is the primary, which is backwards alphabetically, I know, but that's the way it is. Okay, so IgM is a pentamer. It's a very large antibody, and it, like I said before, it also it also fixes complement, which we'll talk more about in a second. Okay, IgD, the only thing you really need to know about it is that it functions mostly as a B cell receptor, and its other functions are largely unknown. That's kind of what it's known for. It's just being a B cell receptor. A lot of other things aren't really known about it. And then IgE is the antibody that is mostly known for allergies and parasitic infections. Okay, so IgE antibodies, for whatever reason, respond to those 2 specific scenarios: allergies and parasitic infections. Okay. All right. So what do these antibodies do? We know that we produce them. So when your B cells and your helper T cells find an infection, they find something that shouldn't belong in your body, they cause the B cells to become activated and produce these plasma cells that act as antibody factories and pump out millions and trillions and however many copies of these antibodies. Why? What is it that these antibodies do? And they do a couple of different things. They have 5 different ways that they affect pathogens. You have to know all 5. Okay. So the first thing that they do is they do something called opsonization. Yes, I can write it down over here. And opsonization is basically just coating a pathogen, as you can see here, so that they can be phagocytized more easily. The antibodies act as like a little flag on the outside of that pathogen that say, hey, this thing is bad, the immune system decided this is bad, and it calls over the phagocytes so that they'll get rid of it. Okay, remember that these antibodies are specifically designed to bind to the receptor or to bind to the antigen that they are specific for. So these pathogens are covered in those antigens. And so those antibodies are going to bind to the outside of those pathogens because they're covered in the antigen that they are specific for. Okay, so opsonization is just the process of the antibody coating the pathogen so that it is more likely to be phagocytized. It's just kind of a signal to the phagocytes to say, hey, this thing needs to go. Okay, the second thing that antibodies do is neutralization or neutralizing. So in neutralization reactions, the antibodies fill up, or they basically block certain parts of the pathogen that make them not able to be pathogens anymore. So specifically, in things like viruses have to bind to your human cells in order to get into them to cause infection, right? That's necessary for them to be able to make you sick. If it's covered in antibodies, the virus won't be able to do that. So the antibodies binding to the outside of that pathogen neutralize it. It's like neutralizing a poison, okay? It makes it so it does not make us sick anymore. Neutralizes it. Okay, the third one is agglutination. And agglutination is a fancy word for clumping. And so what that does is the antibodies will bind multiple pathogens and do what we call cross-linking. It's like a single antibody will grab multiple bacteria and hold them together. And the idea behind this is that pathogens that are clumped together, agglutinated, are easier for our body to get rid of. So it enhances their phagocytosis, and it also makes it more likely that like your organs will get rid of it, like your spleen will be able to see it and get rid of it. Okay, so agglutination is going to enhance phagocytosis. So agglutination is just grabbing lots of pathogens and sticking them all together. Now, a little bit of a side note, I want you to think about the structure of these antibodies and how many binding sites they have. So a monomer has 2 binding sites, right? It's got 2 arms to grab things. Which of these antibodies do you think is the best at grabbing stuff and agglutinating? IgM, because it's got the most arms, right? It's got 5 individual monomers. It's got 10 places where it can grab stuff. Okay, so IgM is the best agglutinator. Okay. And then the last one that we're going to talk about, there is a 5th one, but we're not really going to talk about it. The 4th one that we're going to talk about is complement activation or fixation. Okay, activation, fixation mean the same thing. And remember that there are only 2 antibodies that do complement activation / fixation, IgG and IgM. But what it does is, and again, we're not going to talk about complement much, but we talked about complement in the innate immune system. And complement is just a series of proteins that destroy pathogens. And that's all we're going to say about it. So antibodies that fix complement activate these proteins that will then go on to kill pathogens. Okay, and then the last one is antitoxin, but we're not going to talk about that. Okay, so that's what antibodies do. In all of these different ways, antibodies lead to the death of pathogens. It's either going to cause them to be phagocytized, it's going to cause them to be picked up by the spleen and gotten rid of, it is going to activate complement, which then goes to destroy pathogens. All of those things happen when antibodies are produced. Okay? Opsonization, neutralization, agglutination, and complement fixation. Which again, all of these things are leading to pathogen death. All right, so this is that same image that we looked at in the beginning, and I just put it here again so you could see it again. But these things, again, the B cell side and the T cell side are very interlinked. They happen at the same time. So stage 1 is just the production of those B cells and T cells wherever they're produced. This stage 2 is antigen presentation. Okay, so the antigen presenting cells will consume a pathogen or any kind of foreign object, and they'll present the antigen on the outside of the cell. A helper T cell will become activated because of that antigen presentation, and then will go on to stimulate B cells. And then stage 3 and 4 are once the B cells, once they are activated, they produce plasma cells, which produce antibodies, and they produce memory cells, which remember that infection. And then on the T cell side, that activated helper T cell produces cytotoxic T cells. There's also memory T cells, and these also lead to pathogen death. Okay, there's a lot of steps that happen. But the ultimate purpose of all of this is the death of whatever was invading our body. Okay. I know that was a lot. You may have to watch it a couple times. Okay, so the overall immune response, when we're looking at all of that stuff that happens, stage 1 through 4, all of that together is considered the immune response. So I want to look at a chart now of what really happens to antibody titers or antibody concentrations as it goes through this immune response. And we're breaking this down into primary response and secondary response, which we talked about when I just talked about the antibodies. And the primary response is simply the first time you come in contact with a pathogen, and the secondary is the second time. Okay. So the first time your body experiences a pathogen, it is considered the primary response. The second time, it's the secondary response. So what we're looking at across the bottom is time. And what we're looking at across the y-axis is the amount of antibody. Okay, and notice that we're looking at 3 different things, but mostly we're looking at the red is IgM and the blue is IgG. Now remember that IgM is the biggest one and the first one to respond in the primary response. IgG is biggest in the secondary response, like we said on the chart. Okay, so what happens during the primary response? The biggest thing to realize here is in the primary response, the first time you are exposed to an antigen, so that's what AG is, that's antigen. So the first time your body comes in contact with a new pathogen, a new antigen, the time it takes, think about all those steps we just talked about, the B cells and the T cells being created, the B cell finding that pathogen and presenting a cell, another dendritic cell finding that pathogen and presenting the antigen, a helper T cell becoming activated, this activated helper T cell coming over here to the activated B cell and then creating antibodies. That takes time. It's a lot of steps. A lot of things have to happen in order for the immune response to actually happen. And so there is this latency period between coming in contact with the antigen to when your body is actually able to respond. And the amount of time that can take is incredibly variable, but it can take anywhere from 5 to 20 days, which is a long time. So it can be up to, almost a month from when you are exposed to a pathogen to when your immune system starts to produce antibodies. Okay? Because again, the B cell becoming activated, the helper T cell becoming activated, those two things meeting up, that takes a lot of time. So the first time you come in contact with a pathogen, it takes your body a long time to start producing antibodies, which is why you get sick. Because in all this time, this huge latency period, this 5 to 20 days before your body is producing antibodies, that pathogen is multiplying inside your body, and causing you to become sick. It's having a party because there's nobody stopping it. Okay? And so eventually, over time, the antibodies will go back to normal. The antibodies will be produced, and then the pathogen is killed because of the antibodies and the cytotoxic T cells and all that fun stuff. They will be eventually killed, and you will go back to normal. Now, however much time later, right, this can be any amount of time, from 2 weeks to 100 years, you will come in contact with this same pathogen or same antigen a second time. Now, if you ever come in contact with this exact same pathogen a second time, notice that there is absolutely no latency period here. Your body sees something, when it sees it a second time, it's like, I know this guy. We're not messing around with this. And it immediately, because of those memory cells, start producing antibodies right off the bat. There is no latency period. And so your body immediately starts cranking out huge amounts of antibody to get rid of that pathogen before it has the chance to make you sick. Okay? Notice that the response is much faster, so there's no latency period at all, and the response is much bigger. Look at how much antibodies you produce the second time, or the first time versus the second time. Okay? So the takeaway here is that the second time you come in contact with a particular pathogen, your body will respond immediately and much, much bigger than it did the first time. And this is the whole principle behind why you don't get sick with the same thing twice. Okay? So if you have chickenpox when you're 4 years old, you're never going to get chickenpox again. Do you think it's because you never come in contact with chickenpox again? No. It's just that your body remembers it, and if you ever come in contact with it again, it immediately takes it out. Because it's like, I know this dude. We got to get him out of here. It sucked last time. We're not doing it again. Okay? And this is the whole principle behind vaccines. So vaccines are a way of inducing the primary response without actually having to get sick. So vaccines are a way of introducing antigens. We're going to talk about a couple of different types, but a way of introducing an antigen to make your body go through the primary response so that when you come in contact with that pathogen or antigen for real, because it's trying to make you sick, your body already remembers. And so if a vaccine works perfectly, and they don't always work perfectly, they're not foolproof, but if a vaccine works perfectly, you will never get sick with that pathogen because your body remembers. And you jump immediately to the secondary response instead of starting with that primary response with the huge latency period where you have to get sick before your body can figure out how to get rid of it. Okay? All right, so now we're going to talk just a little bit about adaptive immunity and vaccination. I talked about it just a little bit here, or a second ago, but we're going to talk a little bit more about vaccines, just however much we can cover before the end of the class. So adaptive immunity, there are 4 different types of adaptive immunity. You can have natural and artificial. Natural is what your body does when you are sick. So natural immunity is from getting sick. Or from getting sick. You can have active natural, which is what happens when you personally get sick. Your body has an immune response and produces memory cells that creates immunity. Okay. You can also get passive natural immunity, which happens through pregnancy and breastfeeding for the most part. Okay, because the antibodies produced in the female in this situation, or the person that's breastfeeding, or the person that's giving birth, can be passed through a placenta or through breast milk into the infant or the fetus. Okay, so those antibodies are created in the person who was sick, and they're passed on to their infant or fetus. This is why there's actually really cool videos and pictures. You can look up a lot of this online of what, like the difference between, there's a visible difference in breast milk in a person who is sick and a person who is not. So when a person who is sick pumps breast milk, produces breast milk, it turns like a golden yellow color, and it's because it's full of antibodies, because their body is producing tons and tons of antibodies, which the infant then consume and helps them become naturally immune. Okay. Now the other type is artificial immunity. So this is immunity produced purposefully through medical procedures. The most common type of artificial immunity is active artificial, which is vaccination. It is artificially introducing a pathogen or a part of a pathogen into your body to make your body produce an immune response. And then passive artificial immunity is something called immunotherapy. And immunotherapy is where you can get basically injections of antibodies from other people. So there was a little bit of this during COVID, when COVID was killing so many people. It was possible to get basically infusions of blood from people who had had COVID before. And so the people who had had COVID and survived produced those COVID antibodies, and you can take their blood and inject it into somebody else. Okay, so again, the passive immunity is, it's not something you're producing yourself, it's something you're getting from somebody else. Okay. So early vaccination, the very first true vaccine was developed in 1796, which is a long time ago, by Edward Jenner for smallpox. And I think it's a really cool story, and I think it's one of those things that just reminds you that sometimes it's the simple answer that's the best one. But he noticed, so the very first vaccination was developed by Edward Jenner, and he noticed that dairy maids, for whatever reason, back in 1796, there was a position called a dairy maid, and a dairy maid was a person who milked cows and delivered milk to people. That was their whole job. So they spent a significant portion of their day around cows. And cows have a version of smallpox called cowpox. Now, cowpox is not deadly in humans, typically, but it's very, very similar to smallpox. The antigens are very, very similar. So he noticed that these dairy maids who spent all their time around cows, for some reason, never got smallpox. For whatever reason, he couldn't figure it out at first, but he'd eventually figured out it was because they were all getting cowpox first. Because they spent all their time around cows, they got cowpox, which induced their immune response and then protected them from smallpox. And so he figured out that you could introduce small portions of cowpox into a human being, and that would protect them from smallpox, which was an incredibly deadly infection. It still is. I mean, it doesn't exist anymore, but if we all got smallpox, we would be wiped out. But smallpox, the very first smallpox vaccine, which is kind of a crazy one, and you can actually still be vaccinated against smallpox. They basically don't do it anymore, but the smallpox vaccine for a long time is they take basically antigens. The first ones that they did was they took crusts from the sores of people who had cowpox, right? Because smallpox creates these sores that burst open and create like nasty, crusty stuff. And so they take those crusts and put it on the outside of somebody's skin and then use a needle to poke holes in their skin. So just a little bit of that bacteria would get in there and cause an immune response. And so there are people, and smallpox, people used to be vaccinated for smallpox until it wasn't that long ago that they stopped vaccinating for smallpox. And people who had been vaccinated for it, you can see there's a scar. That's how they used to do it until they stopped vaccinating. They have a little round scar where somebody put antigen on the outside of their arm and then poked holes in it with a needle until the virus got in. So that was the very, very first vaccination, was the smallpox vaccination. And there are different types of vaccines. You can have whole organism vaccines, which is when the vaccine contains the entire organism. Okay, so a lot of whole organism vaccines are like the flu vaccine, the MMR vaccine, things like that. They introduce the entire organism to the human body, and your human immune system produces an immune response to it. Now, there are two different ways that you can do whole organism one. You can do killed, which means that exactly what it sounds like. It's a killed whole organism. And one example of that, the killed cell one, is the flu vaccine. So that is still how most of our flu vaccines are created. Your flu vaccines are created, they grow tons and tons and tons of the flu virus, usually in chicken eggs, but they grow tons and tons of flu virus, and then they kill it all and put it in injections and inject it into your body. Because even though that flu virus is dead, it still has the antigens that your body can recognize. So they inject you with killed organism. Or you can be injected with attenuated organism. And attenuated means that it has been altered in a lab to not be pathogenic. Okay? And so, for example, one example of this is the MMR vaccine, like the MMR, the measles, mumps, rubella. And in that vaccine, there are live versions of measles, mumps, and rubella, but those viruses have been changed so that they don't make you sick. That's what attenuated means. It means they're alive, but they're not going to make you sick. They've been changed. Okay? And then another way that you can create a vaccine is by just using an antigenic component. So this is just injecting part of an organism. Okay, so whatever or part of the organism is the one that's going to induce the best response inside of your human body. And there's lots of different types of this. The hepatitis B vaccine, most of our COVID vaccines are this, pneumonia vaccines. There's lots of different vaccines that are like this, where they just inject a piece of the organism. And I do want to talk specifically about the SARS-CoV-2 vaccine or the COVID-19 vaccine, because the COVID-19 vaccine was the very first mRNA vaccine ever developed. And this vaccine got a lot of flack, right? We talked about this kind of briefly, I think, in the recorded lecture that I posted, that when the COVID-19 vaccine was developed, there was a huge amount of distrust in the people that were developing it. It was a crazy time. I know you guys were relatively young when that was happening, and so you might not have been too aware of what was going on, but it was a crazy time. People were turning on each other like crazy. But when you take all of the politics out of it, it's pretty amazing that we went from, in February of 2020, there was the start of this brand new viral infection that was very, very deadly. It was killing millions of people. In February of 2020, we had a global lockdown in March of 2020. They developed an effective and clinically viable vaccine by November of that year. That's wild. They went from brand new infection to working, distributed, produced vaccine in less than 1 year. That's a medical marvel, okay? It's amazing. And the only reason that they were capable of doing that is because the SARS-CoV-2 vaccine is an MMR, it's not an MMR, it's an mRNA vaccine. And what that means is instead of injecting a whole organism or a dead organism or anything like that, they injected a small piece of mRNA. And that mRNA, when it is injected into our human bodies, our cells pick up that mRNA and produce this spike protein that the COVID-19 virus produces. And that spike protein then goes on to induce an immune response. Now, the reason that's cool is because there's no live virus. There is absolutely zero risk of becoming infected, okay? So there's no live organism. And because there's no live organism, it is cheaper, easier, and faster to produce than most every other vaccine on the market. So all of the other vaccines require the production of, or the cultivation of whatever organism you're trying to inject. Like I said, the flu vaccines, they grow trillions of copies of the flu virus inside of chicken eggs. It takes weeks. And then they have to harvest all of those, they have to clean them, they have to kill them, and then they can make the vaccine. But because PCR exists, you can take one copy of this COVID-19 virus, do PCR on it a bunch of times, and now you have a vaccine. And that's it. And that is why the COVID-19 vaccine, again, you may not have been super aware of this, but after the COVID-19 vaccine was produced, the first one, they had a new one the next February when the Delta variant came out. And then there was a new one 4 months later when the Omicron variant came out. They were able to change it as the virus changed because producing mRNA vaccines is so fast and cheap. In contrast, the flu vaccines, they have already started producing the flu vaccines that will be available next October. Okay, they started producing those back in November probably, because it takes almost a year to produce the flu vaccines for the entire country. Almost a year. That's why a lot of times the flu vaccines don't work, because they're going off of data from almost a year before. So mRNA vaccines are truly an amazing medical discovery. And I hope that as we move forward, there's going to be more that are mRNA vaccines and fewer that are live viruses and things like that. Like I said, they're cheaper, easier, faster. And so far, it seemed to show that they work better. So the mRNA vaccines, from all the data that's been collected since 2020. Now, again, in terms of scientific data, 6 years is not that long, right? The vaccine has only been available for 5 years. So in terms of scientific data, that's not that long. But in that 5 years of data, the mRNA vaccine is shown to be better and to work better than most traditional vaccines. The biggest disadvantage to the COVID-19 vaccine and mRNA vaccines in general is public opinion. The truth is that it's still a relatively unknown process. Like I said, there's no point in downplaying that. 6 years, 5 years of data is not very much. There still could be things that could pop up that we would have no idea about for another 5, 10, 15, 20 years. And so it makes sense that people are wary of something that we don't understand or we can't have enough data for. But as of right now, for the mRNA vaccine that has been produced, the only one that's been produced, the only downside that has been reported is public opinion. It doesn't create any more side effects than any other vaccine, at least that have been reported. So just something to think about. The mRNA vaccines are kind of the new thing, I guess. And hopefully more mRNA vaccines can be produced because they're faster, easier, cheaper. Maybe this is finally the answer to getting a malaria vaccine. Maybe this is finally the answer to getting a tuberculosis vaccine that works well, right? All of these vaccines that haven't been able to be produced in the last 100 years or so, maybe they can now because mRNA vaccines exist. right, and we're just gonna skip this one. But yeah, don't forget your test is on.